Absorption of carbohydrates. Absorption of carbohydrates, in carbohydrates only monosaccharides get absorbed in the intestine. So whatever polysaccharides that we are eating should be digested and broken down to their monosaccharide form, because only monosaccharides get absorbed in the intestine. So the absorption rate is maximum for galactose. Out of all the monosaccharides, absorption rate is maximum for galactose, it is moderate for glucose, and it is minimum for fructose. And as glucose is polar, it cannot diffuse through the lipid bilayer of the cell membrane. So cell has a lipid bilayer and glucose cannot diffuse or enter into the cell. So for the transport of glucose into the cell, there are special glucose transporters, and these glucose transporters range from GLUT1 to GLUT7, where there is GLUT6 is absent, and a detailed video on glucose transporters has been made, which will be given in the iCard or the description box. And these transporters are transmembranous proteins which span the width of the membrane, that is, they will be extending to the whole bilayer of the membrane. Now these glucose transporters are essential for the glucose to enter into the cell from the circulation. So absorption of carbohydrates, mainly there are two mechanisms for absorption of carbohydrates. One is active transport, specially it is a type of secondary active transport, and this active transport mainly occurs from the intestinal lumen to the intestinal cell. So from the intestinal lumen, this is the intestinal lumen, and from here to enter into the intestinal cell, active transport mechanism is needed. This is located on the mucosal side. Now next type of transport in the absorption of carbohydrate is facilitated transport or facilitative diffusion. This is present on the serosal surface. This particular transport is present on the serosal surface, and it helps in the transport of glucose from the intestinal cell to the circulation. So with the combination of these two transport mechanisms, the glucose molecule from the intestinal lumen is transported across the intestinal cell to the circulation. Firstly, active transport. This helps in the transport of glucose and galactose, especially out of the monosaccharides, glucose and galactose are transported with the help of active transport. And as discussed, it is a type of secondary active transport. Now why this is secondary active transport is because the energy utilization occurs indirectly. So active transport, there are two types of active transport mechanism. One is primary active transport and secondary active transport. So if the energy utilization occurs directly, it is called as primary active transport. If the energy utilization occurs indirectly, it is called as secondary active transport. Now here the energy utilization occurs indirectly. How? We'll see in the next slide. So that is why it is called as secondary active transport. Now for this secondary active transport, a special type of carrier protein is required, which is called as sodium dependent glucose transporter. Sodium dependent glucose transporter. Now this sodium dependent glucose transporter is a type of SIM port mechanism. SIM meaning same, port meaning direction. So both, as the name suggests, sodium dependent glucose. So glucose depends on sodium for its transport, and both of them will travel in the same direction. Glucose depends on sodium and both of them will travel in the same direction. Now once they have entered from the intestinal lumen to the intestinal cell, the sodium will again be absorbed into the circulation with the exchange of potassium. So sodium will be expelled outside and potassium will be entering into the cell. Now during this process, there will be utilization of energy with the help of sodium potassium ATPase. So though energy is not getting utilized directly here in the sodium dependent glucose transport, so though the energy is not directly getting utilized here, energy is getting utilized here for the expulsion of the sodium from the cell into the blood. So that is why this is called as indirect utilization of energy, which is otherwise called as secondary active transport mechanism. Secondary active transport mechanism, and it is a type of SIM port mechanism where both sodium and glucose are entering in the same direction. Now the glucose that has entered into the intestinal cell. Now there is one clinical, there are two types of SGLTs, that is sodium dependent glucose transporters, SGLT1 and SGLT2. SGLT1 is present in the intestines, help in the absorption of the monosaccharides from the food, whereas SGLT2 is present in the nephrons, proximal convoluted tubules of nephrons, and helps in the reabsorption of glucose. So whatever glucose that is entering into the glomerular filtrate will be reabsorbed so that it is not excreted by the SGLT2 transporter. So one clinical application of this SGLT transporter is this SGLT helps commonly in the treatment of diarrhea. Oral rehydration solution is given. Now this ORS solution contains glucose and sodium. For the effective absorption of glucose, it is given along with sodium, so that sodium in the salt replenishes the body sodium level, whereas glucose provides energy. So ORS is the common treatment of diarrhea. Why glucose and sodium are given in combination is because if it is given in combination, it helps in effective absorption. It helps in effective absorption. Both of them will be carried through the sodium dependent glucose transporter. So any defect in SGLT1 will cause glucose galactose malabsorption, because these are the monosaccharides which mainly depend on this SGLT transporter. And defect in SGLT2 will cause congenital renal glycosuria. Glycosuria is presence of glucose in urine. Presence of glucose in urine is called as glycosuria. So if there is SGLT transporter defect, the glucose will not be absorbed by the kidneys. Glucose will not be absorbed and it will be coming into the urine. Next is facilitative transport. So the glucose that has entered into the intestinal cell through the sodium dependent glucose transport will be expelled outside with the help of these transporters called as glucose transporters or GLUTs. Now this is a sodium independent facilitative diffusion where there is no energy required. Energy is not required. It's just a facilitated diffusion process, and it operates on the membrane facing capillaries. This is present on the membrane facing or the capillary surface, and it is a uniport mechanism. Uni means only in one direction glucose will be entering from the cell into the circulation, and it is a type of ping pong mechanism of transport. It is a type of ping pong mechanism of transport. So this is a type of ping pong mechanism of transport. So this is what is ping pong mechanism, where the transporter will open when the glucose molecule will come to the surface, and it will catch the glucose molecule, and once it will close towards the luminal surface, and then it will open towards the capillary surface, and because of the opening the glucose molecule will be expelled outside. This process is called as ping pong mechanism, and there are different tissues require different glucose transporters, and as already discussed, glucose transporters will be is already discussed in a special video that will be given in the iCard. And these glucose transporters especially transport glucose, fructose and mannose. Especially for fructose, there is one specific transporter called as GLUT5, which is usually transported by the help of this fructose is usually transported with the help of this GLUT5. So glucose and galactose, they are transported with the help of sodium dependent glucose transporter, and they enter into the intestinal cell, and from the intestinal cell GLUT helps in transport into the circulation, whereas GLUT5 fructose is directly transported from the intestinal lumen into the intestinal or the circulation with the help of GLUT5. So that is what is facilitated transport and the active transport mechanism. Active transport is SGLT or sodium dependent glucose transporter for the transport of glucose and galactose, whereas facilitative is with the help of the glucose transporters, GLUTs, especially for fructose, glucose and mannose.